have only 2 more seminars after today. Is that sad or not? Steve, what do you think?
It's very sad, very sad.
But I have also good news. We will restart in September. So today we have a fantastic speaker and scientist from Germany. He's director of research related to vision. So he is one of our retina fanatics. So hopefully we all enjoy his research today. And with that, I would like to ask Marco to do introduction.
Welcome everybody, ladies and gentlemen. Welcome to our distinguished speaker series of today. My name is Marco Bassetto and I'm a postdoctoral fellow here in the Professor Kaiser Lab at CTV-ARC. It is my true pleasure to introduce our speaker of today and one of my former mentors, Dr. Marius Ueffing. Dr. Ueffing is the director of the Institute for Ophthalmic Research and co-chair of the Center for Ophthalmology at the University Medical Center of Tübingen, one of the largest centers of ophthalmology in Europe. Also, Dr. Ueffing directs the Medical Proteome Center, a core facility of the Medical Faculty in Tübingen, and acts as a co-chair of QBIC, the Quantitative Biology Center of the University of Tübingen. He is also a member of Roche Pharma for the Science and Strategy Advisory Board. Dr. Ueffing has a long-standing history in medical research, as well as background in protein science and molecular and medical genetics. His previous position included that of a research associate at Columbia University, College of Physicians and Surgeons, New York, followed by a group leader position in the pharma industry, and in a division of medical genetics at the University Medical Center in Munich, Germany. Between 2015 and 2019, Dr. Ueffing has coordinated the EU Horizon 2020 System Medicine project named IRISC, focused on AMD, and it was funded with 6 million euros. He published about 290 original articles so far, and he has an H-index of 72. Dr. Ueffing has developed research strategies to combine bioanalytic, proteomic, functional genomics, and computational research towards investigation of disease mechanism and markers. His effort impact the clinical ophthalmology towards understanding and targeting mechanism of disease. He has contributed to the identification of molecular mechanisms of disease as perturbation of protein networks in the brain and neuroretina, the identification of biomarkers for human neurodegenerative disease, and the identification of genes, proteins associated with neurological and retinal disease. He further combined bioanalytical and computational approaches towards better diagnostics and therapies for retina, neurological, and neurosensory diseases, with a strong focus on uncovering molecular mechanisms leading to neurodegeneration. This enabled protein-based diagnostics as well as identification of disease drivers and biomarkers based on the mechanistic understanding of functional protein networks. Dr. Ueffing discovered BCP as a new target for intervention into unfolded protein response stress in retinal neurons and developed a proof-of-concept therapy for retinal degeneration. His seminal work on disease mechanism has initiated development of therapeutic strategies to treat 2 neurological diseases on the basis of molecular constraints. One is an antibody intervention, and the second is pharmacological use of small drug molecules. This work is still ongoing, and thus, without any further ado, I warmly invite you to follow Dr. Ueffing's lecture, which is entitled System Pharmacology for Therapy of Retinal Degeneration, Targeting Proteostasis, the P97 BCP Story. Thank you.
Hey Marco, thank you very much for a very kind introduction. So I may share the screen with you now. Is that okay? And start. I think you can see me, right? Okay, so I hope you can see me. Can see the screen?
Yes.
Great. Okay, thanks again for, I think I have to get this away. Okay. Thanks again for giving me the chance to share your work with you all. It's actually such that I'm still trying to move on. This is how it works. So as you're all aware, I think, retinal degeneration is clinically and genetically very heterogeneous. And so one attempt is actually to go via a mutation-specific approach to treat retinal degeneration. The most notable and clinically now tried approach is supplementation gene therapy. And the other one would be the search for a mutation-independent way of treating retinal degeneration. Here in Tübingen, we do both. We do actually go for a mutation-specific approaches, and we do investigator-driven clinical trials, gene therapy trials, and we also go for the mutation-independent way. And I would like to share with you a kind of approach that is mutation-independent, targeting a protein called VCP. So first of all, before talking about specific treatments, we must understand the disease that will determine which type of therapy might or might not be successful. And so, as I said, retinal degeneration is not one disease, it's a spectrum of many different diseases. And you could do that doing a fishing expedition and just see what is actually going on and be actually successful by chance, or you do this way. You know where to go and you do it in a targeted fashion. And we have decided to do it the way that is depicted here and try to find out how to do it best by actually gain of knowledge. So there has been an approach in the past which was called genome-based pharmacology, where the genetic defect has been in the center of attention, and gene-based drug discovery and pharmacology has been developed. Supplementation gene therapy is such an approach, and it does work in some cases, but in many cases, and especially when using drugs, it hasn't worked well. So the first-in-class development of new drugs has actually decreased and went down with the advent of genome-based pharmacology. Meanwhile, we have a different concept, which is called systems pharmacology, where we do consider the genetic defect, but we do actually consider more. We do actually go from genetics into pathway analysis, and from there into pathway modeling. And from here, we may end up in target definition validation if we are lucky. And we accompany that all by multimodal systematic sets of content-rich data and mechanism-based AI and statistical modeling. I will show you a little bit of that today. And once a target has been validated, we may consider to go into preclinical proof of concept, and from there into clinical trials and hopefully personalized treatment at the end. So in systems pharmacology, the basis are content-rich data. They may be clinical, they may include omics data, and a must is functional data. From there, you generate a hypothesis, which may be true, you validate it, may also be false, you falsify it, and in pharmacology, there's a rule, if you want to fail, fail early, because the more you do, the more expensive it gets. Drug development is expensive, and so you may want to cut down as early as you can. In case you are lucky and you find the data that give you the hope to eventually get a right target, you want to model and validate the pathways where the target is part of, select the right target, then select the right model system to study the disease. Unfortunately, in ophthalmology, we have quite a number of very good models which represent the human disease situation quite properly. You generate a preclinical proof of concept, and most of my talk will focus on that today. And then you plan and go for a clinical trial if you have a solid proof of concept, where safety and treatment modalities are a big issue. And at the end of the day, you want to do a phase 1/2 study where you have to actually include the right patients and in the best case, do personalized treatment. So if we go to the eye and if we look at the eye and if we look at the retina, we have to consider that the retina is the most energy dependent part of our body. And that may be also part of what we have to consider if we think of retinal degeneration. It's not only mutation, it's the systems context of the retina that we have to consider. And especially this part here is extremely energy dependent. So if we move through a photoreceptor, then we are all aware there's a huge packing of outer segment disc in the outer segment. There is the part where the transport is happening that builds up the discs eventually, and there's the inner segment, which is the factory, which is also including a lot of mitochondria that produce the energy that is needed, but also a lot of quality control on the level of protein expression and protein modification that is happening. And this, of course, is the center of gene expression, which also has to be considered. So, as I said, the energy demand in photoreceptors is extreme. The demand for protein folding is also extreme. Also given that rhodopsin and conopsins are GPCRs, which are hard to fold. So the protein folding capacity is at its limit. Moreover, we produce 10% of outer segment discs new per day. And that means if you have a situation like in rhodopsin where you have to face more than 100 different mutations, and we focus on the P23H and the isoleucine 256 deletion mutation. If you have a mutation, it's a mutation in a protein that is extremely highly expressed in photoreceptors. So the demand for controlling protein quality, having a protein like rhodopsin, which is so much expressed and which is vulnerable to mutation, is very high. So what happens if you have a misfolded protein? Then actually the protein folding demand is higher than the protein folding capacity, because as a consequence of your mutation, folding gets more difficult, and folding may never come to an end, because the mutation may affect the proper structure of the protein. And so the machinery may run into many cycles of attempts of refolding before giving up. And this, at the end, may be part of why the photoreceptor degenerates. So moreover, if you think of a molecule like rhodopsin, that molecule has to be transported through the connecting cilium, has to be actually assembled properly to form the outer segment disc. And it's not only a protein that is essential for light reception, it's also essential for actually creating the structure that is making the outer segment disc. And so a whole machinery of different proteins is responsible for doing it right, including quite a number of small GTPases that actually relate the protein complexes to be made. Interfragmental transport as a part of the transport machinery to move the protein and the protein complex, including rhodopsin, to the outer segment, and the machinery, including the cytoskeleton that takes care of the proper assembly of the outer segment disc. So a lot of things can go wrong, and most of the things that you see here are energy dependent processes. So if you have a factory where you have something that goes wrong, you know, you may have this kind of lady, and you may have this kind of lady. And if you have this situation, you know, you may end up in a situation where the whole system breaks down or the factory gets bankrupt. And this, in analogy, is happening when you get into photoreceptor degeneration. So if you consider that the left lady is the inner segment, then building up misfolded protein may lead to having a situation where less protein is actually arriving in the outer segment and more protein gets stuck in the inner segment, which isn't good for the health of the inner segment. So we have actually taken a systematic approach of isolating the outer segment and by sucrose gradient, and then moving into a native solubilization protocol by sucrose gradient, getting native fractions of the outer segments and passing them through a mass spec approach. And this is actually work that is actually was published more than 10 years ago in molecular systems biology, where we took an approach to first actually deassemble the outer segment and then by computational modeling reassemble it by modeling the protein interactions within the outer segment based on rhodopsin in the center, annotating the interactome functionally at the end. And that created a knowledge gain, which we do use for the last 10 years for many different studies, including pathway analysis and analysis of retinal degenerations. Oops, this is the wrong direction. So one kind of peculiar thing that we found, and this is actually from the times where I ran gels myself, is that the highly aggregated rhodopsin, which is shown here on being separated by a native blue native gel, where the first dimension is native blue native and the second is SDS page, where we find valicine containing protein, a protein that I initially didn't know and I didn't expect in the retina, tightly bound to rhodopsin. And this was the start of looking for VCP, valicine containing protein. And so it became clear that VCP is part of this control machinery, keeping balance between protein folding demand and protein folding capacity. And so what happens if protein folding capacity is actually not able to cope with protein folding demand is you run into ER stress. And this is actually where VCP comes in. VCP is a molecular machine, a small molecular machine that works together with a lot of different adapters and cofactors to detect misfolded proteins, especially membrane proteins like rhodopsin. And with the help of cofactors, tags it with ubiquitin, unfolds it, and delivers it to the proteasome for degradation. This works nicely as long as protein demand and protein folding capacity, but also proteasomal degradation capacity is in tune and balanced. If it's not, then the consequence is ER stress, and excessive ER stress can lead to photoreceptor cell death. First of all, there is excessive retrotranslocation, and we see that with rhodopsin in the inner segment, it builds up, and there may be excessive degradation, and all these processes consume a lot of energy, including the activity of VCP, which is in AAA ATPase, which is actually a strong consumer of ATP. So there is work on VCP, and it's not for the sake of retinal degeneration, but it's for the sake of cancer. So a whole range of compounds have been developed to inhibit VCP, mostly to actually interfere with proteasomal balance in cancer cells. And we have used mostly three compounds, irastatin, ML240, but also NMS873 to inhibit VCP. And in the next 20 minutes, you will see the results of that. So in the first part of that study, I will actually share with you data that deal with the pharmacological interference of ML240 and NMS873, two of these inhibitors in retinal axon in the retina with respect to the VCP-ARAT proteasome axis. And to go there, we actually isolate the retina, we generate retinal explants, and we are able to culture these explants for 14 days, sometimes 3 weeks. And we can use these explants for drug-related studies as high content assays. And we have done that with ML240, but also with NMS873. And we've been using them for high content assays, but also for a systematic analysis of protein signatures and pathway analysis in response to the drug addition. So the experimental flow looks such that we treat ML240 as a selective VCP inhibitors for a given time, 2 hours, 6 hours, and 48 hours is a protocol that we used. And then we pass these 4 different explants into mass spec using a tribrid orbitrap fusion machine, which is able to analyze up to 4,000 proteins, identify them and quantify them. And we use these data as a kind of initial data set for further analysis. So VCP is doing many things related to degradation control, protein quality control. And you see here a whole array of functions that are either directly or indirectly coupled to VCP activity. And so what you expect if you interfere with that, you expect a multiplicity of reactions in the cell. And in fact, we see that there is no difference between control and ML240 treatment in a situation where the drug is given for 2 hours. You can see that here. These two situations are almost identical with respect to the fold difference, decrease or increase of protein amount, but also with the, this is actually a log function of the amplitude of change. There's not much happening. This is different after 6 hours. You can see that there is both with respect to proteins decreasing, increasing and decreasing, that there is a apparent difference evolving. And with 48 hour, the fold difference with respect to the heights of protein expression is also increasing. So there's a kind of increase over time. So in concordance to that, if you go into data analysis using an unsupervised clustering AI algorithm, you can see that between control and the 2 hours time point of ML240 treatment, there's not much different. They cluster together. This is actually changing after 6 hours. You see here, there's a cluster on its own. And after 48 hours, there's a cluster on its own, which shows that the drug is doing something, which is actually not only changing single proteins, but also functionally clustered protein landscapes. And this can be also seen in an independent analytical approach where we use UMAP clustering. where relative log intensities are shown here. And you can see between control and 2 hours, there's not much different. And you can see that just comparing these two images. But after 6 hours and 48 hours, there is a difference happen. So clusters that are comparably lowly expressed here change into being highly expressed and vice versa. So there's a lot happening. And the question here is, what are these proteins? What is the nature of these proteins? Do they correspond to specific clusters of proteins? And here, again, using an unsupervised similarity clustering method, we can see that, in fact, there is a change concerning proteins that are linked to retinitis pigmentosa and cone and fundus dystrophy. So there are patterns that change that are disease-related. There are also patterns that are related to other ophthalmological situations like blindness, cataract, diabetic retinopathy. There's a cluster of proteins changing that is related to neurodegeneration, and this is quite a big cluster. And so these heat maps all show protein abundance sorted by hierarchical clustering. The next question is, what are these proteins? And here you see some of them which are actually associated with retinal degeneration, and with drug use, the expression of these proteins goes down. There is a cluster of proteins that is associated with proteasomal degradation, and also with drug use, this goes down. And there is a cluster of proteins that is related to interferon response, and this cluster goes up. This is hard for us to interpret. This may be an adverse effect that is actually cancer related, because in studies where VCP inhibitors are used in the context of developing cancer therapy, we see that in these studies, interferon response patterns are seen to go up. If you put those patterns into pathways, and here you see one of the major pathways that is affected is ER, endoplasmatic reticulum associated degradation related. It's all around ER stress, ERAT, handling misfolded proteins, and unfolded protein response as a consequence of misfolded protein presence, and ERAT. And this is also what we see by classical means. If we stain for single proteins like BIP, for example, a marker for a stress marker for ER stress, we see a similar pattern of reactivity. So next is what happens on the level of the retina with respect to any rescue effect. And here we use the P23H animal, the rat, as well as mice that we study together with Mike Cheatham's lab in London, where we ask, can interference with VCP activity rescue the explants and later the animals from retinal degeneration. So on the explant level, we follow a protocol where we prepare the organ culture, we have a medium change, we may fix the explants, or we may keep them for a longer time to treat them either by ML240 or NNS873, or lately also to other inhibitors, and then fix them and look at them. Or alternatively, actually do intravitreal injections in the intact animal. And here you see one of our early results. If we treat ML240 in retinal explants, we see a lesser abundance of tunnel positive cells. And you can see that here with ML240, but also with Ares-13. And we see an increase in cell rows with both substances, most prominently with ML240. And you can see that here also, you can see the apparent change of more than up to 2 rows or more, sometimes more than 2 rows that we can actually see in difference of the vehicle treated situation. Moreover, if we treat by DMSO, which is a carrier solvent for ML240, we see this pattern where a lot of rhodopsin is actually being retracted in the inner segment, and the Blue Native gel has told us that most of this rhodopsin is formed in aggregates. And with treatment here, it's NMS873, you see that this is almost free of rhodopsin in the nuclear layer and the inner segment. It's all nicely arranged here in the outer segment. Moreover, what we can see in vitro with explants is here you see a vehicle-treated wild-type explant, so no mutation. Here you see a vehicle-treated, DMSO-treated P23H explant. You can see the difference. Here you see a lot of vesicular structure. There's no ordered outer segment disc assembly. And treating these explants with ML240, we can almost get back to this situation, not perfectly. There's still some of the vesicles staying, but this is much better than this. And we can also see that on the level of the outer segment disc length, which is actually coming back. And this is also happening in vivo. If we do a intravitreal injection, then this is actually the untreated situation. This is the treated situation. We see a much nicer and more structured assembly of the outer segment. So this implies that the drug is not only changing the pathways, it's also changing the ultrastructure of the outer segment disc. And the next question to ask is that does it also improve the function of the retina that is treated such way? And this is shown here. This is again NMS873. This is after fixation. Again, you see more cell rows. You see a better distribution of rhodopsin. And we can also observe a better scotopic ERG, both with respect to the A wave and the B wave. And this after treatment with NMS873. So there is a functional improvement, not only a structural improvement. These retina does much better in vivo. These mice are much better off in vivo, and these are data that were actually achieved or obtained in a blinded fashion in Mike Cheatham's group. He didn't know what the vehicle was and what the NMS sample was, and we blinded the study by purpose to actually create unbiased results. And in both our labs, we have similar results. So a single intravitreal injection can protect these eyes for up to 20 days. So why do we think does it work? We think it works because we can restore trafficking such that we can stabilize the outer segment. You may have seen that on the electron microscope level. We get reduced aggregation in the inner segment and a better transport to the outer segment of the visual pigment. We see modified ER folding and UPR response. So we see a lesser UPR response in the treated animals and the treated explants. And we see a decrease in balanced energy consumptions, which we currently measure using the seahorse method. And we see, and I have not included that, but because it has only been done once, we see a healthier mitochondrium in the photoreceptor on the level of electron microscopy with the denser crystal structure, which implies that the mitochondria do better, could do better, but it's only done once, so I don't want to show that because it's preliminary. So the next thing, if you go into drug development, is to formulate your drug. And here, using an approach which is microencapsulation, we could actually show that, and this is a collaboration with Stefano Salmaso's group in Padova, we could show that if we encapsulate ML240 into PEC choline vesicle or cholesterol vesicles, but you will later see that the choline vesicles do much better, that we can actually improve the performance efficacy of the drug and lower the concentration of the drug to be used in our experiments. And again, we do the same protocol. We actually use retinal explants in the first place, treat the explants, and later on we do intravitreal injections. What you can see here in organotypic culture is that the PEC choline alone doesn't make a big difference for them with respect to, in comparison to the not treated situation. But the PEC choline encapsulating or containing the ML240 makes a big difference. You can see that here. This is also quantified on the level of cell rows and the decrease of cell death monitored by tunnel analysis. So this also works in a long-term situation where we've kept these explants for 21 days being treated, and you can see again that the number of cell rows is increasing, can be quantified. The level of cell death is decreased. We have an underlying retinal degeneration due to the fact that the explants don't live forever. So this effect is less pronounced than the short-term treatment. But it shows that the drug is working over time, and it also shows that we don't get apparent adverse effects, which would actually counteract the beneficial action of the drug. So we don't see apparent cell toxicity. The survival profile is actually much better with PEC choline, so the number of dying cells gets down, and this is significant, whereas the PEC cholesterol doesn't do as well. And we have reasons to believe that this is due to the fact that the cholesterol increases the microglia activation, which is almost absent here when we treat with PEC choline ML240 formulation, which is so far the superior formulation over all other ways of delivery. So to summarize that, the ML240 delivery in PEC choline enhances cell survival in the explants. We've started to inject this formulation into animals, and we see a similar effect after intravitreal injection, but this is very preliminary, so this is why I don't show it. We see that there's a restoration of rhodopsin distribution, reduced immune response also compared to the DMSO drug delivery, and a long-term protection in explants. We have to see whether this is also true for the animals being treated. So in the last part of my talk, I want to share with you another way of interfering with VCP activity, and that is siRNA-based inhibition of VCP expression. So the principle is very simple. You create a RNAi which you deliver in a double-stranded fashion such that the unprotected strand is actually processed within the cell, resulting in a single-stranded RNAi, which is actually protected from exonuclease digestion. This RNAi binds to your target mRNA. This is actually inactivated and eventually cleaved, and you have an inhibitory effect based on the reduction of your protein. And again, we use the same protocol, and we have used a method, and this is the collaboration with Marco in Olivier Selfati's group at OZ Bioscience in the Occutare Consortium, where we have actually developed a reverse magnetofection method, where we've actually exposed the magnetic beads with the RNAi from underneath, and then a strong magnetic plate from above to push the RNAi with magnetic force into the retina. And we can see by analysis that this is actually reaching all retinal layers, which is really neat. And here you can see what it does. We can see that this method is completely or almost completely reducing VCP expression. This is the antibody standing against VCP in a non-treated situation. This is the VCP inhibition. And the magnetofection is much better than lipid-based delivery of VCP RNAi, which doesn't show a lot of effect, whereas the magnetofection is really powerful. And this is also resulting in a better survival of the photoreceptors, which you can see here, looking at the cell rows in the outer nuclear layer. So we also see that we have less tunnel positive cells with this method, which is also quantified and statistically significant, and more cell rows, which you can see here, which is also statistically significant. So by two independent methods, by a drug and by RNAi, we reach a similar effect. Inactivating VCP protects photoreceptors from retinal degeneration. So what's very nice is to see that reverse magnetofection does not elicit activation of astrocytes or microglia or Müller cells. So it's actually neutral to that. So we don't get an adverse effect from there, which is actually suggesting that this method could be further explored as a future therapeutic method to bring RNAi into the retina. It's also such that we get a much nicer, better localization of rhodopsin into the outer segment. So it's not as good as with the ML240, but you can see that there is, compared to this situation, that there is a decrease of inner segment rhodopsin, but more importantly, there is an increase of cell rows. So these explants do much better with respect to cell survival of photoreceptors. So to sum up, dominant mutations in rhodopsin increase cellular stress level due to protein aggregation. VCP participates in quality control of protein folding, so which is in basically a beneficial function of VCP, but given that protein demand, folding demand and capacity have to be balanced, VCP activity can actually contribute to pathology if it binds rhodopsin for long and locks it as a misfolded protein in the inner segment of photoreceptors. So we can show that pharmacological inhibition of VCP by intravitreal injection into row P23H rodent eyes. And I have to say, we can also show that for the 256 deletion animals. It's the same thing. This results in a long-term improvement of photoreceptor survival, improves the retinal structure and function in vivo and in vitro. And so we have a proof of concept now for future neuroprotective therapy for autosomal dominant RP. And we are moving from bench to bedside. And the next step to go is into drug-based therapy development. We have evaluated the pathways. We have our target. We have actually leads evaluated. And in the next couple of months, we get new compounds from a compound library that Cleve Pharmaceuticals and a spin-off of Caltech in Pasadena has generated to improve the selectivity specificity profile of the drug further on. We do have a proof of the biological effect in animals in vivo. Now we actually move from the preclinical phase into clinical research. And here, actually, that's actually the valley of death. Here we need to actually plan for a clinical trial, which is, of course, an important thing to do, but also an expensive thing to do, to go into study in humans. And here, safety and efficacy will be most important in the first place. And so there's a lot to do now. As I said, we are here in a preclinical phase of development. We want to move into a clinical study, and we are on our way. This is actually the first map of how to go, and we hope that we can move into that study by end of the year. So with this, I'm at the end, and I want to thank you for staying with me, and I want to thank all the people working with me. All the work that has been done, except native 2D gel, which I did myself long ago, is actually done by my team. And most importantly, Blanca Arango-Gonzalez, Merve Sen, Bowen Chow, and a couple of PhD students whom you see here on the Neckar and canoeing, and with the support of many collaborators. I've already mentioned Marco, who was here in Tübingen, and that was a very fruitful interaction resulting in 2 publications. We work with Anna Grichuk at Harvard, with Mike Cheatham team in London, with Arto Urti's group in Kuopio in eastern Finland, with Ray and Sue Fenn's team at Caltech, and with Stefano's team in Padova in Italy. And I'm very much looking forward to a discussion. I haven't looked at the time, actually. I don't know. I hope there's time left to discuss. Thank you for your attention, and hopefully we have a little bit of discussion. And please be critical. It's really important to get your critique, your brains into discussing the caveats of this, because that's more important than pleasing. Science is the travel for the truth. So please be harsh and criticize, which would help me to do better.
Thank you very much. This was truly wonderful. And I have excellent news for you. The final question and comments will come from Brother Anand. But before we get to him, we have other questions here. Let's start with the room here with 1 or 2 questions. Maybe Arum first and John.
Hello, my name is Arum, working with Dr. Polczewski. Thank you for a great talk today. I am really excited for the ML240 treatment here. So I have 2 questions for the treatment to inhibit the VCP signaling pathway. So based on the ML240 treatment, it looks like it can rescue the degradation of mutant P23H rhodopsin, but it doesn't really correct the misfolding. So in my opinion, it ended up accumulation of a misfolded P23H rhodopsin as well as other misfolded proteins. So in that case, those isorhodopsins might be toxic. So do you think it's just incorporation of a misfolded rhodopsins to the outer segment will actually reduce the toxicity? Is that why you see the in vivo data from this treatment? Because it's amazing how this ML240 improve the in vivo characteristics of a P23H. And then my second question is, then if you compare with the traditional retinoid drugs, which actually corrects the misfolding and then actually rescues also the translocation to the outer segment, how can you compare this ML240 to the traditional retinoid drugs based on the mechanism of action?
Oh, first of all, I'm really happy about this question because, first of all, we have actually asked this question ourselves, and second, we haven't fully satisfied with the answer. So what we actually know, and these are preliminary data, that your assumption is right, that with the downregulation of BCP activity, the folding problem doesn't get corrected, doesn't get solved. So there is rhodopsin misfolded, and this misfolded protein moves into the outer segment. And so can you still see my screen?
Yes.
I don't know. Yeah, you can. And so we believe that what I circle here, that we have a mixture of misfolded and properly folded protein, and we see, and we can assume that this photoreceptor cannot perform as well as a normal photoreceptor having two intact copies of rhodopsin. So and this is actually just speculation. So the photoreceptor can take that. It doesn't degenerate, at least not in the time frame that we have monitored. We don't know what happens over a longer period of time, but we have reasons to believe that the structural reorganization of the outer segment is actually improving photoreceptor stability and also survival. So this photoreceptor will be most likely less performing. It won't detect light at a single photon level anymore. But I wouldn't care if the rod cell stays alive in humans. You know, it doesn't have to perform. I would even accept the night blindness in patients suffering from these diseases if we preserve rod viability and don't get into secondary cone degeneration. You know, we are in the times of electricity, of electric light. We are not in the Stone Age, so it's possible to have that situation, have a poorly performing rod outer segment, a poorly performing rod photoreceptor, but save the retina from degeneration. That's actually my hope, but it's actually speculation based on the fact that we do see that both these molecules are transported once VCP is absent. It's not such that the P23H or the 256 rhodopsin are degrading. They nicely move to the outer segment, both of these protein forms. And it seems not to matter too much. But that's actually phenomenological. For this, we would, to really prove it, we would have to go much deeper. And now to your second question, can you repeat that?
The second question is, if you compare ML240 to traditional retinoid drugs, because the retinoid drugs actually corrects the misfolding and translocate into the outer segment. So based on the mechanism of action, do you think this ML240 is comparatively better or poor, or do you have any idea about that?
No, actually, honestly, I don't have any idea about that. I don't know whether kind of structural mutations that are due to mutation can be properly folded. So there is a software called Foldex that Luis Serrano at EMBL developed. He's now a director at the Center for Genomic Regulation in Barcelona. I've discussed that with him. And so he can model actually misfolding based on a number of constraints. And of course, you can correct some folding errors, but here I think it's uncertain on whether you can correct a folding error that is as harsh as the deletion of this isoleucine in position 256 or P23H. Even if you model that, you cannot be sure whether this can be completely corrected. So it has to be left open. I don't know. If you would develop a combination therapy where you have kind of a drug that acts like a heat shock protein, like corrects the folding at least to a non-pathological state, and then you have ML240 to prevent aggregation. Maybe that would be even better than just ML240 or NMS873 alone. But that's all speculation. The idea is interesting, but I don't.
All right, Marius, I don't want to cut you off, but we have many questions, so just be prepared. Okay. And we're going to John and then to Alan.
Hello, my name is John. I was actually going to ask you about the physiologic nuances between having a mutant protein, but I think you've answered. So my second question is, do you have any plans for, and you probably get this question a lot, plans for making ML240 potentially more specific for photoreceptors? Because proteostasis.
Yes, we are working on it. So ML240 has a little bit of a tendency to inhibit phosphodiesterase 6. And so we are working with Cleve Pharmaceuticals right now to actually improve the properties of ML240 based on a compound library that includes derivatives of ML240, but also other drugs, other leads that are not related to ML240 to get a even better profile. And also solubility is a factor. So ML240 is very hydrophobic, very hard to solve. We want to do better, and the collaboration with Cleve is meant to get to a better drug.
Okay, Alan and then Henry. You're muted.
How about now?
Perfect now.
Okay, thanks for taking my question. It's a fabulous talk, really exciting. And I wonder, thinking over sort of the biological lifetime of a compound like this, do you think your application or even the specific function of VCP that you're looking at may be different under different conditions or age or the disease status? And would that be important over time? That's sort of one set of questions. And the other is like, have you thought about alternatives to VCP? Now that you discovered that VCP is there, knowing so much about VCP, like have you seen differences in ubiquitination or accumulation of ubiquitin conjugates, the processing of those conjugates? So those are two sufficient questions, I guess.
Thank you for these excellent questions. It's actually such that the pharmacodynamics and pharmacokinetics of both these small molecule drugs is not in tune with the therapeutic effect. They have a lifetime or an ATME in vivo of a couple of hours, but the effect stays for more than 2 weeks. And so we have reasons to believe that the drug is actually trapped or withheld in the retina. And we have data that the melanosomes of the RPE are a reservoir for slow release of the drug. With the mice cells, the ATME is much better. So we have a longer time of activity for the drug. But nevertheless, the effect is so much more pronounced than the ATME, the pharmacokinetics of the drug, that we have to assume that there is a storage phenomenon and that that VCP is a long-lived protein, that the drug sticks to VCP, the off rate is very low, so that the drug sticks to VCP for a long time. Now, can you had a second question? And
Yeah, my second question was more mechanistic in terms of trying to figure out what is the VCP really doing? I mean, are you backing up ubiquitin conjugates? Are you altering the processing of the protein other than or in addition to refolding? And may in fact there be other comparable molecules like other than VCP that you could harness?
Yeah, excellent. It's such that we see a lesser polyubiquitinylation of substrates, including rhodopsin, when we inhibit VCP. So the amount of polyubiquitinated rhodopsin decreases significantly, and we see other proteins being less ubiquitinated. So the proteasomal machinery, the proteasome, is actually gets less to do, gets less work. And whether this is beneficial on long time, we have to see once we do repeated experiments in larger animals. In mice, actually, if you inject more than twice into the mouse or rat eye, you more or less destroy the eye. It's actually, the data get very heterogeneous. You can't do that. The lens sac is actually injured and you don't get reliable data anymore. But we're currently, together with Arthur Urti, trying to create data in rabbits, and let's see what they bring.
Mm-hmm. What about alternatives to VCP now that you've opened up this magical box, you know?
Right, so there are alternatives, and we have actually, but this hasn't been followed up a lot so far. We have found another target of the proteasomal machinery that is actually susceptible. Yeah. But we have actually, I actually screened a lot of drugs that are available in the kind of the obvious drugs like that inhibit ARAT or UPR, and most of them don't work. There's only one that works, and we're currently following that up. And because if this would be a neat target, we would like to patent this. So I wouldn't want to talk about it before we know better, and we have filed a patent application. But there is a second target in a pathway related to VCP.
All right. Thank you, Alan, for good questions. Henry and then Corinne.
Thank you. Thank you, Professor. Really, really nice talk. I'm a former postdoc from Chris's lab currently in the University of Eastern Finland. I'm glad to see that you're working with Arto Urti, because my questions actually most of my questions related to the intravitreal delivery, because it's really difficult with the small molecule drugs. So, but I will pass these questions because I also have something else. So in Chris's lab, we did a study on CB5038, and we found that it's extremely strong PDE6 inhibitor.
Oh yeah,
and yes, so
I know this.
So you probably know this. So
yes.
So my question is, like, because these other drugs also they are VCP inhibitors, so are they structurally anyhow related, and do they possess also PDE6 inhibition or not? This was one question.
Yes.
Then
so
I have just a short technical question, maybe a second one. So I'm interested in the organotypic cultures, and in some instances it would be really nice to have the RPE and the photoreceptor interface working. Yes. And have you tried it in like an eye cup, so that you have the eye cup and the retina attached, like similar experiments?
Yes, I could give another talk on this, because what we have is we have two settings. We have human organoids, which where we, the RPE clumps, it doesn't make this nice monocellular lining in human organoids in the self-assembly process, but we have a retina on a dish protocol, which was developed in Stefan Liebau's lab, who is here in Tübingen, where we can actually put the corresponding RPE to the organoid. It's still a little bit there is a curvature, so we don't have fully flat mounted retina, but we have the RPE attached to the neuroretina. Now, there is a second method, and we have published that a few months ago, where we use human RPE. We have published the method with RPE1, but we are now using patient-specific iPS-derived RPE with primary porcine retina, and we use a visual streak of the porcine retina to look at cones specifically. And so we have the RPE as a kind of feeder layer on the neuroretina on top. And we see that this makes a big difference with respect to the survival of the neuroretinal explant. But we also see, and this is related to my A&D work, that risk alleles like complement factor H, for example, 4 or 2, that they make a difference. They reflect on the neuroretina if we culture the human RPE together with the porcine retina. So these systems are in place here, and we are routinely working with them now.
Okay, Corinne and Tiansen.
Hi, I'm Corinne Gosser from AbbVie. My question was partially answered, but I want to expand a little bit more on it. So VCP plays an important role, and as scientists, we always have to balance between inhibiting something important and keeping it going. So you mentioned some toxicity. Maybe you can expand on that. And have you seen any? You mentioned you've seen adverse events. So how do you think we're going to deal with this in the long run, given the fact that VCP has a real job to do, and would we be harming more than helping?
Right. There is even a kind of hereditary disease actually linked to VCP mutations. So VCP is a necessary component, but luckily this disease doesn't affect the eyes. It's a kind of mostly muscular disease. So that's very lucky. Nevertheless, I believe VCP has an important role, and it may be such that the dose makes a poison. So we have to tune. So these patients would have to be treated like the CNV patients that receive Lucentis or so, anti-VEGF inhibitors. So you may have to treat them repeatedly to have a kind of duration of effect, and we don't know how that will be. It's pretty clear that if we overdose our drugs, we actually get into strong adverse effects. And So the dose makes a poison, and the encapsulation that we do with pecolan is also increasing the safety. We need 20% of the drug that we need without pecolan, and we get a much more stable and long effect on the level of protection.
You mentioned something happened. What actually happened in the second injection to the retina? You said it melted. Like, what exactly happened?
So in the second injection, we cannot use the DMSO ML240 formulation. We have to actually use pecolan formulation, because the DMSO, you know, the anatomy of the rodent eye, you know, it's such a lens and such a little vitreous body. So you get local effects. And if we do, if we sacrifice the animals, and if we look at what happens, we see local effects most likely produced by local overdosing and by local toxicity due to DMSO. And the pecolan is actually giving a much nicer distribution and doesn't give us the kind of local toxicity. There's another thing: if you inject more than 2 times into the rodent eye, the data get very inconsistent. You get response to injury, and you damage lens sac and retina in many cases. So we have abandoned that. We don't do that anymore.
Yes, sir.
Thank you.
Yeah. Yes, hi. This is Tianxin Li at NEI. Very nice talk. My question is related to the very first one. So it seems like the idea of the VCP treatment or inhibition leads to a relaxation of the protein quality control, allowing more of the mutant proteins to go out to the other segment.
Yeah.
That being the case, and years ago, Gordon Fine and John Listman published a equivalent light hypothesis for RP, and that work was followed up by Carter Cornwell and Dan O'Brien, provided some additional support. The idea is that free opsin, not conjugated to 11-cis retinal, activates phototransduction at low efficiency, 6 orders magnitude, but that's enough, given the number of molecules in the rod, to wipe out light response and basically saturate the rod response. And mutant opsins were proposed to have similar properties. So that, given that, do you think your data, first of all, lend any support or even refutation of that hypothesis? I noticed that you found the ERG to be increased, and I was looking for maybe perhaps based on the hypothesis that ERG is suppressed, even though there is morphological improvement. So I want your comment on that, and whether this increased noise, substantially increased noise, random activation for the transduction in the outer segment in the absence of light could have long-term deleterious effect, a side effect, if you will, to the treatment.
Right. It could, for example, result in photophobia or some other things that we can't control in the animal right now. So it's very important what you say, because this outer segment disc is not a normal outer segment disc. So with respect to light adaptation, with respect to crosstalk with the cones, we don't know what happens. That is something that is hard to assess in an animal. And so I would be actually happy to get suggestions how to tackle this problem, which could even be a problem from the first moment of human clinical trial. The mouse wouldn't tell us about photophobia or about anything that is kind of weird due to treatment. And we don't want to really go into that. once we are in the clinical trial. So would you have suggestions how to?
I don't have a suggestion about your specific question, but I was going to suggest that if you use the transgenic mice that have missed species, like earlier transgenic mice were made by introducing mutant forms of human rhodopsin into the mouse background. And with that kind of a host, you could easily tell the mutant opsin whether it actually does get transported more efficiently with VCP inhibition, or maybe there is some other outcomes for the mutant form of opsin. And that could be the first step in addressing this question.
Yes, in actually following that line, we have fused rhodopsin with mCherry and GFP so that we can discriminate both forms of rhodopsin and monitor their kind of transport. So these experiments are currently being done because by antibody, so far we cannot discriminate between the mutant and the wild type protein in the kind of heterozygous dominant situation. And you're right, there is this rat that Master Lavelle actually made, which I think has a human rhodopsin in a transgene being expressed. That could also give us some information.
Okay, let's go to 2 questions from the room. Emily.
Before the question, can I just say that because he was asking if we have any ideas, when you do primate studies, you will find out about photophobia and any other.
Yes, so that's the next thing. So we have the go and the money for the non-human primate study. So that would be the next to do, actually, moving from the mouse to primates. Yeah. Thank you. It's reassuring. Yeah.
Hi, my name is Emily, and I'm a graduate student here at UC Irvine. Thank you so much for your talk. I have a quick technical question that was kind of related to Henry's question about the retinal explants. How do you confirm viability of these explants at, say, 14 or 21 days to ensure that, this model is physiologically similar to what you would find in vivo?
So it's actually the protocol, let me go back, is actually a consequence of more than 10 years of trial and error of Blanca Arango Gonzalez. She's an MD by training, but she's actually mastered this protocol over 10 years. It's actually very much the microenvironment that this explant is encountering, including the medium composition and the medium change, that is actually making these explants viable over time. So it's a kind of tedious protocol, but it is actually quite reliable. We also do that for the porcine explants. It works also for those explants. So it's not bound to a single species.
Thank you.
Jennings.
Okay. Hello, Jennings Liu here from the PhD lab. Really enjoyed your talk. I have 2 questions. The first one is more of a big picture question. So the work you presented primarily deals with the proteomic stress component of retinal degenerations. And I was wondering if you could comment based on your findings of the relationship between the proteomic stress and inflammatory stress components of retinal degenerations. And second, more specifically, with the P23H heterozygote model you showed, where a single intravitreal injection was protective. I was wondering what age those mice were, and if you also looked at different ages and the relative efficacy at the different time points.
Yeah, that's also a very relevant question. Thank you for that. So here you can see that these animals are very young, postnatal day 9. That's super young. We have used these younger animals, and we have also used animals where we actually started to treat at PM30. If you do it later, there is a point of no return. So once you have a real dystrophic situation, the VCP inhibition doesn't work well anymore. We are currently expanding our model to, or our approach to animals that are not in degeneration because of rhodopsin mutations, but for example, to RD10, which is actually a PD6 mutation. And preliminary data tell us that there the degeneration starts around day 25, 30. And there we can see that in explants being made from day 20, we also see increased stability and a functional gain of these explants with treatment, with anti-VCP treatment. But it's also that's again very preliminary, hasn't been produced enough to say this is really hard data.
All right, I know everybody's waiting for Dr. Swarup to share his wisdom, but we have a final question from Alan.
Second. Okay, so my question is to change the topic a little bit, going to AMD. Would it be advantageous to enhance VCP or comparable activities to clear out damaged proteins, such as proteins, normal proteins and the accumulation thereof that cause dysfunction in the RPE and cause AMD?
Yeah, then you have to. That's actually that's great that you asked that. So you may have. Yeah, you may have already felt the porcine streak model. We don't do that for RD. We like hereditary RD. We do that for AMD. We take the visual streak to look at AMD patterns, and we're currently trying to feed RPE with outer segment discs to see on whether that creates stress and whether that would act, and whether VCP inhibition would actually create a benefit. So you may be, yeah, you're definitely aware of that. There are two concepts. One on which cells dies first. There, the RPE dies, in fact, but there are also rods dying in the early or intermediate phase of AMD. And so there is a problem with that rods have in coping with the fact that the RPE performs less well because the Bruch's membrane is thickened or the RPE is already compromised. And with respect to this scenario, we're currently trying to find out whether VCP inhibition may be of help. And there is preliminary data that we can protect rods better in the visual streak porcine model when we give a VCP inhibition. But it's too early to say on whether the whole system, the co-culture between RPE and porcine visual streak profits. That's a new project that was just actually granted. It's the student is here now for a couple of months, and so it's too early to say whether it's really working or not.
Thanks.
All right. And now, ladies and gentlemen, Dr. Alan Swarup. He's muted. Alan, you're completely muted. That's unusual.
That's a good news.
No, I actually wanted to mute myself while laughing. You know, I didn't want you to see my laugh. So anyway, so Marius, you know, as you can see yourself, your talk generated just such a wonderful discussion. And there's just so much to unwind here and unfold here, actually, in your terminology. So I actually had, a couple of simpler comments in the beginning, but listening to discussion, particularly starting from question 1, made me think about much larger comments, but I think we have only 5 minutes there. So I think my comment, so I'm not going to ask you question, since I'm visiting you next month, maybe I'll ask questions that time. I think with respect to the comments, there are two. One is based on the dominant mutation and the mechanisms and the way people perceive toxicity. And the second thing is about the toxicity of your drug and the questions that were coming up along the way. I'll take the first one, rather the second one first. With Chris, we helped him with some of the experiments, and we had a couple of nice papers together where we showed that suboptimal doses of different drugs in combination could be better than the right amount of doses of one. And that was based on, Chris, you remember those papers, I'm sure. So I think my own feeling is that if you have a little bit of toxicity, that's okay. I mean, it sort of doesn't hurt. And I want to, I think it can be alleviated in different circumstances by adjusting the dose or not having a complete effect. And you did talk about it in answer to one of the questions. I mean, my philosophy is I've been working on rods all my life. And I say, even if rods don't function, who cares? As long as they're alive. Yes. Okay? As long as they're alive and well, that's what we were showing earlier in some of the other experiments. As long as they can hold the fort, as long as they can stay alive, be there for cones to work and to do whatever they need to do. I think for human beings, you can be in light all day. I mean, you don't need too much light. I mean, actually, David Papermaster used to say that we use rods only when we go out to pee in the night or something. Yeah, I mean, that was David Papermaster. So just basically, bottom line is, as long as rods stay alive, they keep the fort, they hold the fort, I think we are fine. So I think that's that. Coming back to the dominant mutations, I mean, in 1984, we published probably one of the first papers on dominant mutations in Drosophila, where we proposed that the mutant protein is going to scavenge the wild type protein. It was a P150 glued mutation in Drosophila. And a lot of papers have talked about dominant proteins can be toxic or this or that or something. There's no single hypothesis going to be or mechanism going to be in any of those cases. I think you can alleviate dominant mutation just by simply overexpression of the protein, as we just showed in case of CRX, dominant LCA in organoid models, where you have mutant protein, which is supposed to be toxic and all that stuff, but you overexpress wild type protein and that can rescue the effect partially. Even if it doesn't do fully, who cares? Okay, you can see partially, I'm fine with that. So I think in the big scheme of things from patient perspective, as long as you can alleviate whichever way. Now, there could be multiple ways to look at cell death. Trafficking, block is one, but trafficking block itself is not a very simple part that you're talking about, and the ER stress. I remember papers from, I think it was Al Lewin, Marina Gorbachev, and other Gorbachev and others talking about BIP, overexpression of BIP that can alleviate ER stress in P23H. So I think there could be many different ways, and the mechanism for your drug is probably going to be, rightly as you are saying, not unfolding the protein or not doing any properly folding the protein, but probably making a right path for it to go through. Maybe it just opens up ER a little bit wider, it relaxes, like, and it's so that, all right, the mutant protein, which is not well folded, can now pass through and go wherever it is going and may not work very well. So I think there could be many different ways dominant mutations can exert their impact on cellular pathways and eventually lead to cell death. We tend to be reductionist, and we just sort of listen to one thing that, okay, this is how it is, and everything, has to fit in that format. I am very open-minded. I'm happy to take any hypothesis, and they all may be right. They don't need to be mutually exclusive. So I think in my mind, I think we need to think of multiple ways to solve a problem rather than trying to fit everything in one box. So I think
it was wonderful, Alan, and thank you very much for a fantastic presentation today, great conversation. And I wanted to finish with something positive. So we will invite Dr. Swarup for next year to speak as well again. Thank you very much.
Thank you.
Right. Thanks, Marius.
Thanks, Anand. Looking forward to see you soon.